Ali. I'm a PhD student at the University of Birmingham, studying how our urges and impulses can be affected in Parkinson's disease. This research is funded by the Humane Research Trust, who are celebrating 60 years improving human health through human models, thereby reducing harm to animals for experimentation. Impulse control is the ability to withhold inappropriate behaviours and is heavily influenced by levels of dopamine in the brain. Consequently, individuals with Parkinson's disease have a higher risk of developing impulse control disorders such as binge eating, gambling and shopping addictions. These behaviours present severe consequences for the patients and their families, yet there is currently no objective way of identifying individuals who are more at risk. Therefore, the purpose of our study is to examine measures at the level of the muscle and the brain which may be related to impaired inhibitory control. In this video, we will demonstrate the use of brain stimulation and muscle activity recordings to examine inhibitory control in a movement task.
Our participants will perform this movement task, intercepting 2 filling bars at a target line by lifting their left and right index fingers off 2 switches. However, on some trials, the bars stop rising automatically. In these trials, the participant is required to not lift the corresponding fingers. The ability to prevent this pre-planned movement is an indicator of impulse control. During this inhibition task, we will use the technique called surface electromyography to record the activity of the index finger muscles. Whenever the muscle contracts, a burst of electric activity is generated which can be recorded from neighbouring skin areas. Participants will also receive a type of non-invasive brain stimulation called transcranial magnetic stimulation. When we stimulate the motor cortex, the brain region which controls our movements, we can produce contractions in the hand muscles on the opposite side of the body. You can also see the results of stimulation in the electrode recording. Shortly after stimulation, a motor evoked potential, or MEP, is observed in the finger muscle. The MEP amplitude provides a measurement of the excitability of the pathway between the motor cortex and the muscle, with a higher amplitude MEP signalling greater excitability. Therefore, we'd expect MEPs to get larger when participants are preparing to move their fingers and to get smaller when the participants must cancel their movements.
We predict that people with Parkinson's disease will show abnormal patterns of muscle activation and a reduced ability to withhold their prepared finger movement, as their drive to the muscle will remain high throughout the task, reflecting impaired inhibitory control. We will also investigate whether changes to muscle and brain activity recorded in this task are associated with impulse control issues in everyday life. This line of research is working towards developing a tool which indicates individuals who are at a higher risk of developing an impulse control disorder. Thank you for listening.
Thanks to the Humane Research Trust for supporting this research.